Everyone. I am fortunate enough to introduce Dr. Rati Tripathi, Assistant Professor, Department of Pharmaceutical Science, Susruta School of Medical and Paramedical Science, Assam University, Silchar, Assam. She has around 10 years of teaching and research experience. Dr. Tripathi has received Senior Research Fellowship from the Indian Council of Medical Research during her PhD. In addition, she has received a research fellowship from the University Grant Commission during her PhD. She has during her post-graduation. Area of expertise of Dr. Rati Tripathi is design of novel small molecule by rational structure-based drug design and ligand-based drug tetracycle, semicarbazole, and hydrazone. Funded by Indian Council of Medical Research, Government of India. In addition, she has published 13 research papers in national and international peer-reviewed journal. In addition, she is the author of book entitled Fundamental of Pharmaceutical Inorganic Chemistry, published by IK International Private Limited, New Delhi, and Essential of Pharmaceutical Inorganic Chemistry, published by Ahuja Publishing House, New Delhi. So with all due respect, I would like to request, ma'am, please enlighten us on computer-aided drug design. Over to you, ma'am.
Thank you, Ishan sir. Good evening, everyone.
Thank you, madam. Namaskar.
For such a very nice introduction. Yeah,
Thank you,
so
madam.
first of all, I would like to thank the Parul University and the organizers of this webinar, the principal of Parul Institute of Pharmacy, Dr. Abhay Dharmasi sir, for providing me the opportunity to share my knowledge with you all. So let me just share my presentation with you.
Yes, ma'am.
So I hope that all of you are fine and taking good care of yourself in this pandemic situation. So moving further, the topic of my today's talk is computer-aided drug design. We know that drug design, it's an inventive process of finding new medications based on the knowledge of a biological target. It involves the design of molecules that are complementary in shape and charge to the biomolecular target with which they interact and therefore binds to it. Introduction of new therapeutic solutions is an inventive and time-consuming process. As you can see, this is a drug discovery and development timeline, and you can see that it has been estimated that a typical drug discovery cycle from lead identification to clinical trials, it takes around 14 to 15 long years, with the cost of around $800 million per marketed drugs. In the early rapid developments in the field of combinatorial chemistry and high throughput screening technologies have created an environment for expediting the drug discovery process by enabling the synthesis of huge libraries of compounds and screening them in short periods of time. So this is what is called as combinatorial chemistry and high throughput robotic screening. However, these concerted efforts not only failed to increase the number of successfully launched newer molecular entities, but seemingly aggravated the situations. Hit rates were found to be very low, and many of these identified hits failed to further optimize into actual leads and preclinicals. So among the late stage failures, 40 to 60% was reportedly due to absorption, metabolism, excretion, and toxicity deficiencies. Moreover, huge infrastructure and logistics was required, not only for the screening process, but also for the preliminary synthesis, storage, and maintenance of compounds. So collectively, these issues underscored the need to develop alternative strategies that can help remove the unsuitable compounds before the exhaustion of significant amount of resources. So in this time, a new paradigm in the drug discovery came underway, calling for an early assessment of potency, activity, and selectivity of the lead candidates, as well as their potential ADME tox liabilities. This assisted to reduce costly late-stage failures and accelerated successful development of new molecular entities. At the core of this paradigm shift is the application of computational techniques to facilitate the discovery of new molecular entities. So thus, computer-aided drug design, which can be shortly called as CADD, that is CAD, it is a widely used term that represents the computational tools and resources for the storage, management, analysis, and modeling of compounds so as to facilitate the design and discovery of new therapeutic solutions. It also includes development of digital repositories for the study of chemical interaction relationships, computer programs for designing compounds with the interesting physicochemical characteristics, as well as tools for the systematic assessment of potential lead candidates before they are synthesized and tested. The more recent foundations of CAD were established in the early 1970s with the use of structural biology to modify the biological activity of various proteins. At that time, X-ray crystallography was quite expensive and time-consuming, rendering it infeasible for the large-scale screening in the industrial laboratories. So this figure, shows the various stages of the modern drug discovery and development process. As you can see, it consists of various, steps like identifying a disease, isolating a protein which is involved in the disease, finding a drug which is effective against the disease protein, preclinical testing, formulation and scale up of the lead which has been obtained through the preclinical testing, and then followed by human clinical trials and then FDA approval. So it is at this third stage, that is when we are trying to find out a drug effective against the disease protein, that CAD, that is computer aided drug design, comes into play. Over the years, new technologies such as virtual screening, molecular modeling, and in silico ADME tools have emerged and begin to be exploited in the lead design. So as you can see that it is in between the second and third stage, various types of computer aided drug design tools are involved, which helps us to find out new therapeutic solutions in shorter period of time. That is, they will help us to reduce the number of molecules synthesized. So one of the CAD methods is virtual screening, in which we use a computer to predict the biological activity of the designed molecules. We have molecular modeling processes wherein computer graphics and models help us to improve the activity, help us to modify the activity of the molecules. And we have in silico ADME models, which helps us to predict the ADMET properties of the designed molecules. So this entire figure, it gives you an idea about the involvement of various advances of like genomics, proteomics, high throughput screening technologies, and computational infrastructures to bridge the gap between the theoretical modeling and medicinal chemistry. So CAD nowadays plays a very critical role in the search of new molecular entities. As you can see in these arrows, you can see the yellow colored and the orange colored arrows gives you an idea about various CAD activities which are nowadays utilized in the drug discovery process. Current focus includes improved design and management of the data sources, creation of computer programs to generate huge libraries of pharmacologically interesting compounds, development of new algorithms to access the potency and selectivity of the lead candidates, and design of predictive tools to identify the potential ADME tox liabilities. Thus, the objectives of CAD is to change from random screening to rational drug design and testing. Let us now have a look into the major tools and resources that have been developed for expediting the search for novel drug candidates. Molecular modeling, as you can see here, molecular modeling, it's a part of computer-aided drug design. So molecular modeling encompasses theoretical methods and computational techniques to model or mimic the behavior of molecules. It is important to note here that the common feature of molecular modeling methods is the atomistic level description of molecular systems. So we focus upon the atomistic level of the molecular systems. We study the atomistic level, we study the molecular systems on the atomic level. It involves a range of computerized techniques based on the theoretical method and experimental data to predict the molecular and biological properties. So molecular modeling in drug design is performed by two most common methods, that is molecular mechanics and quantum mechanics. Both these methods produce equations for the total energy of the structure. Further, molecular mechanics consists of two important drug design techniques, that is structure-based drug design and ligand-based drug design. Structure-based drug design is also called as target-based drug design. In structure-based drug design, three-dimensional structure of the target is known. Please note here, target can be either the receptor, it can be an enzyme, it can be an ion channel, or it can be nucleic acids as well. So in this case, in case of structure-based or target-based drug design, we are well aware about the three-dimensional structure of the target protein. While in case of ligand-based drug design, the three-dimensional structure of the target protein is not available or it is not known. So one of the very important approach of structure-based drug design is molecular docking, while QSAR studies and pharmacophore modeling is a part of ligand-based drug design. Depending upon the knowledge of three-dimensional structure of the target, you can choose any of these techniques to design and identify novel therapeutic molecules. Now let us see what can be done if we are aware about the three-dimensional structure of the target protein. So if the three-dimensional structure of the protein is known to us, then we can identify the active site within the protein by studying the binding site surrounding the bound co-crystallized ligand. And then we can further proceed with the docking studies to ascertain the binding pose and interactions of the ligands within the active site of the protein. If the three-dimensional structure of the target is not available or it is not known to us, then we can go for homology modeling of the target protein. Homology modeling is also known as comparative modeling of the protein. In homology modeling, an atomistic model of the target protein is constructed from its amino acid sequence, and an experimental three-dimensional structure of the related homologous protein is constructed is used. We can search or identify the binding site in the homology model. We can specify the binding site, and then we can proceed with the docking studies. Now, before proceeding further, let us have an overview on the molecular mechanics and quantum mechanics. So molecular mechanics is a more popular method compared to the quantum mechanics. And the reasons behind this is that The process is quite simple to use and it requires less computing time to produce a model. It has to be noted here that the molecular mechanics is based upon the total potential energy of the molecule, which is given by the sum of all the energies of attractive and repulsive forces between the atoms in the structure. So as you are well aware that any molecule, it consists of atoms which are joined or which are linked together by the bonds. So we can have various type of interactions between these atoms and the bonds which are present in them. So you can see we have torsional bonds, we have torsional angles, we have angle bending, non-bonded interactions, and bond stretching. So these various types of interactions which are present between the atoms and the bonds plays a very important role, and we focus upon the calculation of potential energy of various parts of the molecule in molecular mechanics. That is, in other words, we can say that the total energy is considered to be the sum of Taylor series expansions for the stretches for every pair of bonded atoms, and it also adds on additional potential energy terms for the angle bending, torsional energy, and some non-bonded interactions, which may include van der Waals energy, hydrogen bonding interactions, maybe electrostatic interactions as well. So these energies are calculated using the mechanical model in which the atoms are represented by the balls. As you can see in the figure here, the atoms are represented by the balls whose mass is considered to be the proportional to their relative atomic masses. And the atoms are considered to be joined by the mechanical springs. These springs correspond to the covalent bonds which link the atoms within the structure. So you can see here various types of interactions which are involved between the atoms, and the bonds includes electrostatic attraction, van der Waals interaction, angle bending interactions, dihedral rotations, bond stretching interactions. So all of these various types of interactions and their corresponding energies will contribute to the total potential energy of the system. So the calculations are based on two important considerations. That is, the isolated structures are considered at 0 Kelvin temperature, and secondly, it does not take into account the effect of environment on the structure. So thus the total energy of the system may be expressed mathematically by the equations. Please note that these equations are known as force fields. So this is the equation which has been shown here. It represents the total potential energy of the system. Which is the summation of the stretching energies, bending energies, torsional energies, van der Waals energies, and electrostatic interactions. So this is all about molecular mechanics. Now let us move on to quantum mechanics. So in contrast to molecular mechanics, quantum mechanics provides information about the nuclear position and distribution of electrons. It is based upon the wave properties of electrons and all the material particles. The mathematics of the wave motions are applied to the electrons, atoms, and the molecular structure. Quantum mechanics is basically the molecular orbital calculation, and it offers the most detailed description of the molecule's chemical behavior. Please note that it takes into consideration HOMO and LUMO properties of a molecule. Quantum mechanics is based upon the Schrodinger's equation. That is the Schrodinger's wave equation. So this is the Schrodinger wave equation, which is expressed by H psi is equals to E psi is equals to U plus K psi, where psi indicates the wave function. It's a time-dependent wave function which describes the electron distribution around the molecule. The capital E, it represents the energy of the system relative to one in which all the atomic particles are separated to infinite distances. That is, it is equals to total potential energy and total kinetic energy of all the particles in the molecule. This capital H represents the Hamiltonian operator that operates on the molecular orbital, or that operates on the wave function side to determine the energy. So quantum methods utilize the principles of particle physics to examine the structure as a function of electron distribution. Quantum mechanics is useful for calculating the values of ionization potential, electron affinities, heat of formation, dipole moments, and other physical properties of atoms and molecules. It also calculates the relative probabilities of finding the electrons. That is, it helps us to calculate the electron density within a structure, and it helps us to assess the nature of binding of a possible drug to a target site. So these are some of the applications of quantum mechanics. So this slide, it's a summary of molecular mechanics versus quantum mechanics. So as we have seen that the quantum mechanical methods or quantum mechanics deals with the electrons in a system, the calculations has been found to be very time consuming. In contrast to quantum mechanics, the molecular mechanics or force field methods, they have been found to ignore the electronic motions. So electronic motions are not taken into consideration in case of molecular mechanics, which is based upon the principles of classical physics, while the quantum mechanical methods are based upon the principles of particle physics. So this is the main difference between these two. And because of this, molecular mechanics calculates the potential energy of the system as a function of nuclear position only. The calculations has been found to be very less time consuming. So these are some of the very important differences between the quantum mechanics and molecular mechanics. It has to be noted here that the molecular mechanics cannot provide answers that rely on the electronic distribution of the molecule. The reason is that molecular mechanics ignores the electronic motions, and that's why in this case we can use quantum mechanical methods for finding the solutions for such questions. Now, the drug design strategies based upon the molecular mechanics includes structure-based drug design, which is also called as target-based drug design. And docking is a very good example of it, in which we need to have target protein known to us, and we should have ligands to proceed for the docking. And this finally helps us to find out a lead molecule. Another approach of molecular mechanics is ligand-based drug design, in which the target protein structure is unknown to us, but we are well aware about the structures of the ligands. And the most important approaches of ligand-based drug design is pharmacophore and 3D QSAR study, which again helps us to find out the leads. In structure-based drug design, the macromolecular target, that is either the enzyme or protein, under study is to be isolated and then crystallized. And after crystallizing, its three-dimensional structure is to be determined using X-ray crystallography. Nowadays, we have a lot of other techniques which has been used to determine the three-dimensional crystal structure of the target protein. Like we have NMR, nuclear magnetic resonance spectroscopy, and we have also nowadays using electron microscopy to determine the three-dimensional structure of the biological target. But this structure does not give information about the binding site because there is no ligand attached to this target. However, the co-crystallized structure of the biomolecular target complexed with the inhibitor has been found to be very useful for the study. The reasons behind this is that it gives us information about the active site of the enzyme. It also helps us to calculate the distance between the inhibitor and the binding site boundaries. And it also helps us to find out the possible interactions between the inhibitor and the binding site. So thus, the co-crystallized ligand, which is bound to the inhibitor molecule, has been found to be more useful than mere the crystal form of the target molecule. To use the co-crystallized structure, firstly, we need to remove the inhibitor from the active site of the enzyme. Then the protein is subjected to energy minimization to obtain the enzyme or protein in its lowest energy state, because we know that the lowest energy state is considered to be the most stable. So that's why energy minimization is a very important step in this process. In the next step, the lead compounds are docked into the active site of the enzyme to see or to ascertain their binding pose and interactions within the enzyme. Best fit compounds will then be synthesized and tested for the biological activity. Thus, this process will help us to reduce the time for synthesis and screening of large number of compounds. So structure-based, target-based drug design is used to design a new molecule based upon the knowledge of the three-dimensional structure of the biological target. As you can see, we need to have a receptor and we need to have a library of ligands with us, which can be potential inhibitors. So first of all, we need to identify the active site or binding site within the receptor or protein molecule. And then we try to dock these potential inhibitors or ligands within its active site. And then we get the results in the form of scores or binding free energy values. So by analyzing the interactions and these binding free energy values, we can confirm whether the drug can be a potential inhibitor or not. The most widely used approach of structure-based drug design is molecular docking. Docking attempts to find the best matching between the two molecules. Here the two molecules are a ligand molecule, or which can in the future may act as a potential drug, or a receptor or enzyme. Thus, docking is a method which predicts the preferred orientation of one molecule to a second when bound to form a stable complex with the overall minimum energy. Thus, docking is the identification of low energy conformation after binding of ligand molecule within the biological target. The process of docking a ligand to a binding site of a target mimics the natural course of interactions of the ligand and its target protein via the lowest energy pathway. Thus, given the two biological molecules, docking helps to determine whether the two molecules interact or not. If so, what is the orientation that maximizes the interaction while minimizing the total energy of the complex? So these are the two important applications of molecular docking. That is, it helps us to predict whether the molecules interact or not, and if so, what is their orientation within the active site of the protein. The goal of molecular docking is to be able to search a database of molecular structures and retrieve the molecules that can interact with the query structure. There are three types of docking: ligand-protein docking, protein-protein docking, and protein nucleic acid docking, which is sometimes also called as protein DNA docking. So these are the three types of docking. Further, there are three important approaches to the ligand-protein docking. That is rigid body docking, in which the target protein and the ligand both are treated as rigid bodies. In the semi-flexible docking, only the ligand is considered as flexible, while the target protein is treated as a rigid body. While in flexible docking, both the ligands and the protein molecules are considered as flexible molecules. The summary of these three approaches has been presented in this table. Now, let us have a look into some, docking softwares, free and commercial softwares which can be used to perform molecular docking. So examples of free softwares, that is free for academic softwares, includes Doc, Autodoc, Surflex, Fred, and eHeats. And some of the commercial softwares includes Gold, Glide, FlexX, Discovery Studio, MOE, etc. There are not many other docking softwares which are available for use. So till now, whatever we have discussed in structure-based design was on the basis of the known three-dimensional structure of the target enzyme. Now let us see what happens when the crystal structure of the target enzyme is not available or is not known to us. So in this case, we have these three options. That is, we may use the recombinant DNA technology to produce the desired enzyme using the bacterial cell. We may use the homologue of this enzyme from the other organism like bacteria or parasite. So this is called as homology modeling or comparative modeling. Again, this is a computational technique. And we may use ligand-based drug design. So these are the three approaches which can be used when the crystal structure of the target protein is not available to us. In ligand-based drug design, the three-dimensional structure of the target protein is not known or not available to us. However, the ligands are well defined and characterized. Please take a note that ligand-based drug design is based upon the assumption that similar chemical structure may have similar chemical activity. So in ligand-based drug design, we begin with the biologically active molecules. First of all, we try to identify the common chemistry in all those selected biologically active molecules. Then we search or we design new compounds that match or that resemble the chemistry of the compounds identified in the second step. And then we assume that the compounds that match the above description will also be active. So these are the steps in the ligand-based drug design. Please take a note that initial two steps, that is these two steps, involves the process which is called as model building. That is, identifying or beginning with the biologically active compounds and then identifying the common chemistry in those compounds. So these two steps are a part of a process called as model building, while the next two steps, that is the final two steps, they are known as database screening. So in ligand-based drug design, we identify the active compounds from the database and then we search or design more similar compounds. As you can see, we start with the database of compounds. We try to find out the known actives from this database of compounds, and then we design or search for the structures which which shows resemblance with these known actives. In the absence of the structural information of the target, the ligand-based method makes use of information provided by the known inhibitors for the target receptor. One of the very important approach of ligand-based drug design is QSAR, that is quantitative structure activity relationship. We know that QSAR is a mathematical relationship between the biological activity of a molecular system and its chemical characteristics, geometric and chemical characteristics. QSAR is given by the following formula, where activity is considered to be the function of molecular or fragmental properties. QSAR attempts to find a consistent relationship between the biological activity and the properties, molecular properties, so that these can be used to evaluate the activity of new compounds. In other words, QSAR is a statistical approach that attempts to relate the physicochemical properties of molecules to their biological activities. It consists of various types of descriptors like molecular weight, number of rotatable bonds, log P, etc. are some of the commonly used descriptors or parameters of QSAR. So various types of structure properties can be used for the correlation in QSAR. As here, an example has been shown, in which hydrophobicity has been correlated with the biological activity. So in this case, activity of the drugs, we know that it is often related to the partition coefficient. So this figure, it's an example which shows the binding of drugs to the serum albumin. The property, that is log P, is plotted on the x-axis, and the biological activity, which is a dependent parameter, it is plotted on the y-axis, and we see that whether we obtain a straight line or not. So it can be seen from this figure that for a limited range of log P value, we have got a straight line which is expressed in the form of an equation, statistical equation, that is log of 1 by C. That is, this is what the biological activity is. That is, log of 1 by C is the biological activity, and this is related to m times log P plus C. So you can see that this equation can be compared to the straight line equation, that is y is equals to mx plus C, where you can use these data, that is the slope and C data, to see what is the effect of log P on the biological activity of the molecule. Various types of structure properties can be used for the correlation in QSAR. Some of these properties has been enlisted here. So we have chemical properties like pKa, log P, solubility, stability, etc. We have intrinsic properties of the molecule we can use in QSAR, like molecular volume, connectivity indices, charge distribution, molecular weight, polar surface area, etc. And the biological properties which can be included may consist of activity values, toxicity values, biotransformation, or pharmacokinetics. Now, these are the various QSAR results, various types of QSAR results. So you can see that we have various types of QSAR, like we have atom-based QSAR, we can go for field-based QSAR, we can go for group-based QSAR to determine whether the molecules are active or not. The figures are the results of atom-based QSAR, field-based QSAR, and group-based QSAR studies for a particular molecule or for a particular system. And the results are expressed in the form of these plots, which are called as contour plots. So please take a note that the QSAR analysis generates the virtual receptors, and it determines the quantitative relationship between the biological activity of the set of compounds and the three-dimensional properties via statistical correlations. So as you can see, these two figures, the first two figures, they are the results of atom-based QSAR. As you can see here, the contour plots, there are highlights, the blue colored contour plots are said to be good for the molecules, while the red colored contour regions are said to be bad for the molecule. So please take a note that in case of atom-based QSAR, we take into consideration the following parameters like hydrogen bond donors, hydrogen bond acceptors, hydrophobic groups, negative groups, positive groups, electron withdrawing and electron donating groups. So in case of atom-based QSAR, the following features of the molecules are taken into consideration, and we try to identify which part of the molecule is favorable for the biological activity and which part of the molecule is not favorable. So in this figure, the regions which are shown by the blue color, they are considered to be the favorable regions for the biological activity, while the regions which are shown in red color are considered to be the bad regions or unfavorable regions for the biological activity. So such regions, the red colored regions, can be modified to increase or to modify the biological activity of such molecules. These two figures are an indication of the field-based QSAR and group-based QSAR. So in field-based QSAR, we take into consideration the electrostatic and steric fields. As you can see here, the favorable steric interactions are shown by the green color, and the unfavorable steric contours are shown by the yellow color. So please take a note here. You can see here the green colored part. It is a favorable steric contour, while this yellow colored part is an unfavorable steric contour. That is, it indicates that these groups are unfavorable for the biological activity, while the green colored regions, they favor the biological activity. Similarly, this red colored the red colored part indicates the favorable negative groups which are present, while the blue colored indicates the favorable positive groups which are present in the molecule. So this is how it will help us to identify which groups are favorable, which groups which are present in the lead molecule are favorable for the activity and which are not favorable for the activity. So please take a note that field-based QSAR includes only electrostatic and steric fields. That is, as you can see here, favorable, positive and negative charges. And then steric fields are taken into consideration in case of field-based QSAR. While in case of group-based QSAR, we take into consideration not only the steric and electrostatic fields, but we also take into account the various other groups like hydrogen bond donor features, hydrogen bond acceptor features, and hydrophobic features are also taken into consideration in case of group-based QSAR. So you can see the first figure, that is in case of group-based QSAR, the A diagram, it indicates two different colors. You can see here we have a yellow colored region and we have a green colored region. So this is a steric contour basically. So steric contour, the green colored region indicates the favorable region and the yellow colored region indicates the unfavorable region for the biological activity. So this indicates that we need to modify this unfavorable yellow colored unfavorable region to obtain a compound with better biological activity. Similarly, this B figure indicates the electrostatic contours. So here in this case, the blue colored part, it indicates the favorable electrostatic contour, while the red colored part indicates the unfavorable part of the molecule for the biological activity. So we need to modify this unfavorable, that is red colored part of this contour so as to obtain molecule with better effect. Similarly, in case of C, that which indicates the hydrogen bond donor contours. So in this case, the plum color, it indicates the favorable regions present on the molecule, while the cyan color indicates the unfavorable regions, unfavorable hydrogen bond donors present on the molecule. So you can modify these blue colored hydrogen bond donors, cyan colored hydrogen bond donors to obtain a better molecule. Similarly, this D indicates a hydrogen bond acceptor contours, in which the red colored portion indicates the favorable contours, favorable groups, hydrogen bond acceptor groups, while the magenta color indicates the unfavorable groups which are present in the molecule. This E figure in the similar manner again gives us an idea about the hydrophobic contours. So in this case, the yellow colored indicates, the yellow colored portion indicates the favorable regions, favorable hydrophobic groups present in the molecule, while this magenta color region indicates the unfavorable part of the molecule. So by using these, information, we can modify the structures of the molecules so as to obtain better molecule with better biological property. So this is about QSAR. Another approach of ligand-based drug design is pharmacophore modeling. So as I told you that there are two important approaches to the ligand-based drug design. One of them was QSAR. The other one is pharmacophore modeling, which is sometimes also called as pharmacophore mapping. So before we discuss further, let us have a brief idea about what is pharmacophore. So as per the IUPAC, a pharmacophore is considered to be the ensemble of the steric and electronic features that are necessary to ensure the optimal molecular interactions with a specific biological target and to trigger or block its biological response. So in this case, that is in case of pharmacophore modeling, we take into consideration the steric and electronic features within the molecule, which is responsible for the interactions with the specific biological target. And these interactions will then define whether it favors the biological activity or whether it unfavors the biological response. You can see here we have a ligand and we have a receptor. First of all, we try to identify the various regions on the active site of the receptor, and based upon these features, we try to design the ligands which possesses the features which are complementary in shape and charge to that of the receptor molecule. So the various features which are taken into consideration over here is hydrophobic feature, hydrogen bond acceptors, hydrogen bond donors, positive ionizable groups, and negative ionizable groups. Thus, the pharmacophore is considered to be a special arrangement of atoms or functional groups which is required for a drug molecule to exert a particular biological effect. Or in other words, we can say that pharmacophore is the schematic representation of the nature of the biological functional group, bioactive functional groups, along with their interatomic distances, as you can see in this figure, which is present on the right hand side. So you can see that this is the structure of adrenaline, and it consists of various types of functional groups in its structure. So our main objective through pharmacophore modeling is to identify the pharmacophore from this structure. So in this case, you can see here we have identified three important groups or three important pharmacophoric features from the structure, like say for example NH, then these OH groups, and then this phenyl ring. And then we try to calculate the interatomic distances between each of these groups. So this is what is done in pharmacophore modeling. So as I told you that identification of pharmacophoric features is a very important part of pharmacophore modeling. So any pharmacophore can be represented by various types of molecular features. So these are some of the pharmacophoric features which are considered during the pharmacophore modeling. This is hydrogen bond acceptor, which is represented by this color. Hydrogen bond acceptor lipophilic. Please take a note of the size of these groups and their colors as well. So hydrogen bond acceptor, we have hydrogen bond acceptor lipophilic, we have hydrogen bond donors, hydrophobic groups, hydrophobic aliphatic groups, hydrophobic aromatic groups. So these are represented by cyan colors. we have aromatic rings, we have negatively ionizable, positively ionizable groups, we have negative charge and positive charge. So each of these can be considered as pharmacophoric features. So our first goal in pharmacophore modeling is to identify the presence of any of these features within the molecule, which may be responsible for the biological activity of the molecule. So this figure shows a molecular framework. As you can see that this, the structure has been presented in the form of sticks, and various pharmacophoric features has been identified within this molecular framework. So you can see it consists of aromatic ring, which is shown by this orange colored, ball-like structure. We have hydrophobic group, which is present in it, which is shown by this cyan color. We have hydrogen bonding acceptors, which are shown by this blue colored balls. Then we have hydrogen bond acceptors, again by this color. We have hydrogen bond donors, which are represented by this magenta colors. So this is an example of a molecular framework possessing various types of features essential for the molecule's biological activity. This was about the identification of the pharmacophore and the various types of pharmacophoric features which are present in the molecule, essential for the molecule's biological activity. Now let us move on to molecular modeling. So molecular modeling is a three-dimensional computational approach largely exploited in the drug design, mainly to rationalize the distribution of activities within the group of molecules exhibiting a similar pharmacological profile and supposed to be recognized by the same site of target protein. So for this purpose, the very first step is the pharmacophore model that most likely encodes the three-dimensional organization of the required interaction pattern is created. So this is what we have done in the previous, we have seen in the previous slide. That is, we try to create a pharmacophore model or a query structure which helps us to encode the organization of the required interaction pattern along with their interatomic distances as well. The goal of pharmacophore modeling is to establish the bioactive conformations of the ligand and to superimpose the mapping. Secondly, to superimpose the mapping, we need to have the structure-activity relationship of the structurally diverse and conformation in informative molecules. You can see here the figure on the right-hand side, it gives you an idea about the pharmacophore modeling and the various steps which are involved in the pharmacophore modeling. So as you can see that it's an iterative or cyclic process. We can start with the pharmacophore. So if you already have a pharmacophore available for particular, group of molecules or for the particular target, you can use that to search for the compound library for the active molecules, or you can even start with searching compound library for the actives. So either you can start with this step if you have already available pharmacophore model for a particular target. If you do not have the pharmacophore model for a particular target, then in that case, you can start with this step. That is, you can start by searching the compound library for the known actives. Then this is followed by the next step, that is, you can buy or synthesize those hit molecules. You can test them for the particular pharmacological activity, and you can use that activity data along with their structures to generate the pharmacophore. So you can see that we have been able to identify the various pharmacophoric features within the molecule, and then in addition to that, we have been able to identify the interatomic distances between each of these atoms which are present within the molecule, which is essential for the desired pharmacological activity. So these are some of the applications of pharmacophore modeling. You can search for the databases. We can go for scaffold hopping, means you will change the main nucleus of the structure, main part of the molecule. You can modify and try to have a better molecule. We can go for virtual screening, we can go for ligand profiling, we can go for prediction of activity, fragment design, and post filtering. So these are some of the applications of pharmacophore modeling. So that was all about the various tools and techniques, which are a part of computer-aided drug design and which are widely used to obtain novel therapeutic solutions. Now let us have a look into the applications of computer-aided drug design. So drug design with the help of computers may be used at any of the following stages of drug discovery process. That is, it helps us to identify hit by using virtual screening. So we can either use structure or ligand-based drug design for this purpose. So the first application was hit identification. The second one is hit to lead optimization of affinity and selectivity. So for this, we can go for structure-based design or QSAR. And the third application is lead optimization. That is optimization of optimization of molecule for the other pharmaceutical properties while maintaining the affinity and biological activity of the molecule. So these are the applications of computer-aided drug design tools. Now let us have a brief look into the success stories of computer-aided drug design. So various potential channel blockers have been discovered by use of CAD processes, like structural based discovery was used, or structure-based drug design was used to identify potential channel blockers. Some calcium antagonist or calcium channel blockers has also been developed with the help of computer-aided drug design technique. So chemical descriptor based discovery, that is chemical descriptors were used, or in other words, QSAR was used to identify calcium channel blocker. And other example includes the development of anticoagulants like thrombin inhibitors were developed by molecular docking and de novo design. So these are some of the success stories of computer-aided drug design. About the advantages of CAD, so it's a cost-effective process method. It can be used to reduce the time period for the drug development and optimization. The most important is that the molecular graphic technique, which is used in CAD, it will help us to get information about the drug receptor interaction and atom scale binding properties to particular ligand or protein. And this is how it may give new ideas to the researchers to modify the drug compounds for the improved fit. Thus, computational methods have accelerated discovery by reducing the number of iterations required, and in addition, have often provided more novel small molecule structures. Though this technique suffers from a lot of advantages, it does possess some limitations. So ideally, the computational method should be able to predict the affinity before a compound is synthesized. And hence, in theory, only one compound is needed to be synthesized. The reality, however, is somewhat different. These computational methods which are available are somewhat imperfect, and they provide at the best only qualitatively accurate estimates of the affinity. So this is a limitation of CAD-based tools. Therefore, in practice, it takes a lot of iterations of design, synthesis, and testing before an optimal molecule is discovered. So this is all from my side. Thank you, everyone.
Thank you very much, madam. Am I audible?
Yes, sir. Yes, sir.
Yes, sir. So with all due respect, on behalf of Parul Institute of Pharmacy, Faculty of Pharmacy, Parul University, I would like to thank Dr. Rati Tripathi ma'am for a talk on computer-aided drug design. In her current webinar, she has discussed about the computer-aided drug design, molecular modeling, molecular mechanics with force field explanation, quantum mechanics, difference between molecular mechanics, quantum and molecular mechanics, drug design strategy based on molecular mechanics, 3D QSAR, structure-based drug design, X-ray crystallography, molecular docking, and its types. In addition, she has also given the valuable information about the various drug docking softwares. Dr. Rati ma'am has discussed about the ligand-based drug design and its importance in drug discovery, QSAR, types of QSAR, importance of log p-value, steric and electrostatic field, pharmacophore modeling and its features, application of CAD and its success stories. So with all due respect, I would like to thank Dr. Rati ma'am for accepting such nice invitation. I am also thankful to the Dr. Abhay Dharamsi sir, Dean Faculty of Pharmacy, for giving this opportunity to me. I am also thankful to management of Parul University for such a nice online platform for learning, as well as I am also thankful to PU webinar team. Thank you very much. Thank you very much, all of you. Thank you, sir. Thank you, madam.
Thank you, sir. Thank you, sir.
Thank you very much, sir. Thank you very much.
So can we leave,
Okay.
sir?
Yes, yes.
Yeah, yeah. Thank you, sir.
Thank you, sir. Thank you, all of you.